optimization of sampling plan for the different risk targets in milk and dairy products. One of the main objectives of this work package is to identify risk factors for their performance objective and food safety objective values for various categories of milk and dairy products. The assessment of appropriate food safety criteria has been performed for the specific food and risk combinations, which lead us to the following activities. First, it is prevalence, concentration and distribution data which have been collected for Listeria monocytogenes in cheeses and for verotoxigenic E. coli in milk. Second, the effect of transport and storage on growth or inactivation of Listeria monocytogenes inoculated in sheep cheeses and cow cheeses stored at refrigeration and abuse temperature, which have been assessed. Data on temperature changes in storage of fresh and boiled unpasteurized cow cheese and the marketplaces and the retail fridges have been collected. Also, the distribution of VTEC within milk samples has been investigated. What about prevalence of Listeria monocytogenes in cheeses? Experiments regarding the prevalence of Listeria in fresh, semi-hard and hard cheeses made from unpasteurized and pasteurized cow or sheep milk were conducted, and it is shown that the prevalence of Listeria is very, very low. Because of this, we have gathered additional literature data to fulfill gaps for model development. What about growth or inactivation of Listeria monocytogenes inoculated in cheeses? We evaluated the temperature abuse regarding survival of Listeria monocytogenes by inoculation of sheep cheeses with Listeria and to evaluate the fate at different temperatures, refrigeration temperatures 4 and 12 degrees and abuse room temperature. The presence and enumeration of Listeria monocytogenes was done using ISO standards. Also, 3 independent samples were evaluated for the enumeration of lactic acid microbiota using also ISO standard. However, the degree of survival of Listeria monocytogenes was different depending on temperature, while more than 4 logs reduction after 70 days was observed when samples were stored at room temperature. An increased time was needed when samples were stored at refrigerator temperature. For 4 degrees it was 104 days and at 12 degrees it was 95 days. In addition, detection of Listeria but below of limit of quantification, for example 10 colony unit per sample, was achieved after 74 days at room temperature, after 98 days at 12 degrees and 111 days for samples stored at refrigeration temperature. Accordingly, no detection of Listeria monocytogenes was observed at day 77 of storage at room temperature, at day 104 for samples stored at 12 degrees, and at day 122 for samples stored at 4 degrees. Results regarding the evaluation of the effect of temperature abuse on survival of Listeria monocytogenes in sheep cheeses artificially contaminated shows that while the lactic acid microbiota is stable or grow along of the experiment, a statistically significant reduction of Listeria monocytogenes was observed in the free temperatures assay. A systematic review of the literature was conducted to identify the critical factors influencing the growth of Listeria monocytogenes during cheese production. It is evident that the occurrence of Listeria is sporadic and several factors influence pathogen contamination of cheese, including the farm environment, environmental cross-contamination in cheese production, improper processing and handling, and storage prior to consumption. Based on specific literature, framework model has been initiated to model the growth of Listeria monocytogenes and survival at each stage of cheese process. The models predicting Listeria monocytogenes growth showed good correlation for selected temperatures, 4, 10 and 15 degrees. During storage at 4 degrees and 10 degrees, growth of Listeria was observed in pasteurized cheeses, but not in raw cheeses. In the other experiment, boiled cheeses were spiked with independent Listeria monocytogenes inoculums and incubated for 28 days at 4 and 13 degrees. The results collected show that there is an abrupt growth in the first 2 days and a slight increase up to 2.4 log colony-forming units. As no significant differences existed in concentration data at the temperatures tested, it was decided to make another separate experiment for 2 to 3 days with more analytical points to complete this gap in order to generate predictive model. So again, boiled cheeses were spiked with 10 milliliters of 10 to 4 colony-forming units per milliliter of Listeria monocytogenes. Growth of Listeria was measured every 4 hours in duplicates. Rapid growth was observed during first 12 hours after inoculation, with maximum value of approximately 3 logs after 36 hours. There are no significant differences of growth on different temperatures. To estimate growth potential during storage, a challenge test was performed at 4, 7, 10 and 13 degrees during 28 days, which is expected shelf life of the cheese. Growth estimation of Listeria monocytogenes in boiled cheese, assuming dynamic storage, showed the value on which performance objectives should be fixed, not to exceed food safety objectives. The need for quantitative data pushed the partners to develop quantitative PCR methods that could be used as an alternative to the colony counting and most probably number methods. They are too much laborious and time-consuming. Results of studies carried out to validate quantitative PCR methods with a pre-enrichment step were reported. Quantitative and more specific methods allowing lower detection levels are needed, as the PO values in early process stages should be very low for human pathogens that may grow during processing. Our first chosen product risk combination is hard sheep cheese and Listeria monocytogenes as risk. According to baseline results, the establishment of food safety criteria and sampling plans for Listeria monocytogenes in hard cheese is not practical. Expected prevalence in a lot is too low to detect a sufficient number of positive with current analytical methods applied. There are ISO methods. Sampling plans require a high degree of intensity to be able to discriminate between accepted and rejected lots. No information about microbial concentration of Listeria monocytogenes in contaminated samples was obtained. According to the challenge test performed, survival and/or fate of Listeria monocytogenes is expected during the shelf life. It is approximately 96 days. Considering hard cheese as a ready-to-eat food, if the established FSO is 2 log colony-forming units per gram, a very high contamination of samples after production would be needed to cause listeriosis due to cheese consumption. Therefore, sampling plans may be useful as alternative tools to obtain information about the expected prevalence of Listeria monocytogenes in the food lot and how contamination can be distributed within this lot. Nevertheless, it must be pointed out that the intensity required is not practical, so they do not fully serve to discriminate between contaminated and non-contaminated lots. Hard cheese is a food product where the low water activity together with low pH and antimicrobials produce inhibition of Listeria monocytogenes during storage. Thus, if good manufacturing practices are accomplished, the final product may be Listeria free. However, positive samples can appear due to post-contamination or recontamination during elaboration. Basing on these results, sampling strategies might be alternatively addressed to determine microbial prevalence and concentration on food contact surfaces or aerosols in order to localize the main source of pathogen contamination. Regarding the sample size of the analytical portion to be tested, 25 grams of solid food is the general consensus. However, we have demonstrated that the level of Listeria contamination in cheese is low, so the use of higher amount of samples is advisable. Within our work package, we have developed an analytical strategy based in enrichment in the half-racer and subsequent DNA extraction and Listeria monocytogenes specific real-time PCR for the sensitive detection of this pathogen using higher-mouth. We have demonstrated that following this approach, the limit of detection was 1 to 10 colony-forming units per sample, but using samples of 50 grams instead of 25 grams. This increased the analytical sensitivity and therefore could also increase the chance of detecting the pathogen. The next product risk combination is boiled cheese and Listeria monocytogenes. Boiled cheese is defined as a traditional cheese made from non-pasteurized cow milk. For the establishment of food safety criteria and exposure assessment was conducted. The log increase of Listeria monocytogenes at the end of the shelf life was the model output. The expected prevalence of the pathogen was firstly investigated to estimate growth potential during storage. A challenge test was performed at different temperatures during 28 days, which are expected shelf life of the product. To evaluate the effect of temperature, two scenarios were considered. For the first scenario, 50 days of storage at 4 degrees plus 13 days of storage at 12 degrees, growth of Listeria monocytogenes exceeded the FSO at 25th day of storage. This implies that microbial contamination should be lower in the final product or a lower storage temperature may be required. The performance objective could be established just after elaboration, considering the maximum concentration or frequency allowable to fulfill the food safety objectives of two logs at the end of the shelf life. For the worst case scenario, which means 50 days of storage at 80 degrees and 13 days of storage at 12 degrees, this value should be low, 5 colony-forming units in 200 grams. Therefore, higher storage temperatures would need a lesser contamination level to achieve the same food safety objective. To estimate the number of samples needed to accept or reject a lot, a presence or absence test must be done in order to detect positive samples. It can be seen that 4 samples are needed, assuming a storage of 15 days at 8 and 13 days at 12. So the worst scenario, if 25 grams sample is taken. For the scenario with storage temperature of 4 degrees, 5 samples would be required to have the same discrimination power. Nevertheless, it should be pointed out that the probability of detecting positives slightly increased from 10 to 25 gram samples. For the scenario of storage at 4 degrees, taking 5 samples of 8 grams each would produce the same efficiency as 5 samples of 25 grams. Likewise, for the scenario of storage at 8 degrees, 4 samples of 11 grams each would produce the same efficiency as 4 samples of 25 grams. So, to optimize sampling plans, sampling size can be reduced in both cases, since the number of samples to obtain a 5% probability of acceptance is the same. The work carried out in baseline is related to the evaluation of different alternatives for the rapid and sensitive detection of Listeria monocytogenes in cheese. For a complete evaluation of a novel methodological approach, different factors have been evaluated. The inoculum level, 1 to 10 colony-forming unit per samples, 1 to 100 colony-forming unit per samples. Sample size, the standard amount of 25 grams or 40 grams. The dilution, 1 to 10 as standard dilution, but also 1 to 3 and 1 to 5. And the incubation times, standard incubation for the first enrichment of half-racer for Listeria of 24 hours, but also incubation time of 18 hours. And 3 different bacterial DNA extraction protocols. Evaluation of standard and alternative methods, ISO methods versus quantitative PCR, in detection of low number of Listeria in cheese, shows that results obtained using the helix and boiling protocols are quite poor and not reproducible. The results suggest that only Kozačinski columns can be used. Also, results show that PCR is useful for screening potential positive enrichments. Listeria-1 broth method gave more positive than ISO method and needed 1 to 2 days less time. In general, reference methods for detection of pathogen can be time-consuming and expensive. In comparison, alternative methods are rapid, high-specific, sensitive, and expensive in the range of standard methods. As they are not reference methods, they could be accepted from industry as screening methods, and interpretation of the results could be hampered by the current regulations. Our next biological risk was salmonella in cheese. Analysis of pasteurized and unpasteurized sheep cheeses directly from the cheese-making factories on salmonella were performed using ISO methods. No positive samples was detected. Also, detection of salmonella was done using 3 strains of salmonella, similar as for listeria, with 3 different extraction protocols: Helix, Voynich, and Kiev and Colmes. And a specific study for quantification of salmonella W in raw milk samples by quantitative PCR was done. An optimized PCR protocol was developed and demonstrated a good reproducibility for artificially contaminated raw milk samples and detection limit from 1 coliform unit per milliliter. Our third pathogen is verotoxigenic Escherichia coli. The prevalence of verotoxin-producing E. coli and enteropathogen E. coli of serogroups O157 and O26 was determined in 102 bulk raw milk samples and 81 milk filters. In milk, just one O26 VTEC was isolated, which showed verotoxin and EAE genes, and 4 samples allowed detection of enteropathogen with EAE genes isolates of serogroup O26. Other 2 E. coli isolates of serogroups O157 did not show verocytotoxin or EAE genes. Results of monitoring showed that the concentration of bulk raw milk was very low, 4.2 most probably number per milliliter, and the distribution was not uniform. Less than 5% of samples had 2 or more than 2 positive sample units out of 5. As the VTEC serotypes are of major interest for human health, food safety objectives have not been defined, nor are microbiological criteria indicated in EU regulations at the moment, except the absence of O157 serotypes in raw milk, intended for direct consumption. Dairy sampling schemes for VTEC in milk are the regional sampling plans that require analysis of 1 sampling unit. Plans are inadequate because it was observed that prevalence is low and distribution in mass bulk milk is not homogeneous even after mixing. The distribution of VTEC within milk samples has been investigated. A total of 24 over 40 tested lots showed negative PCR results. Moreover, real-time PCR showed that O157 and O26 strains, when quantitative PCR and real-time PCR results show value equivalent to 10 to 3rd coliform unit per milliliter. For isolates belonging to the O157, O26, O103 and O145 cell groups were found to have significantly similar D values at 58 and 62 degrees. The next product and this combination is raw milk and VTEC. Sampling for microbiological risks in milk is currently performed according to EU regulations. Official sampling schemes for VTEC are still missing, and it seems crucial to include in future sampling protocols a sensitive and specific analytic method for both detection and quantification of this pathogen. PCR methods could be used as an alternative to the colony counting and most probably number methods, which is too much laborious and time-consuming. For the detection of VTEC in milk, isolates were obtained from the samples when quantitative PCR and real-time PCR results show values equivalent to 10 to 3rd or above. Therefore, this is a value that should be reached in the enrichment broths in order to allow confirmation by using cultural methods. As alternative methods are not reference methods, they could be accepted from industry as screening methods. Sampling frequency should be harmonized between countries. Also, the regional sampling plays that require analysis of one sample units are inadequate. Appropriate sampling schemes for raw milk intended to be sold raw to final consumers need to consider that the distribution of beta in milk is not uniform and concentration is very low in positive samples. Raw milk can be sampled at farm level within 24 hours of storage at 6 to 8 degrees. The next product risk combination is E. coli and mozzarella. Experiments were done to evaluate the survival of verotoxigenic E. coli of serogroups O157 and O26 during the mozzarella stretching phase. The data show that after a first sharp decline of most probably number counts, it was approximately 3 logs, reduction within 1 to 2 minutes. There is an evident tail where most probably number